Hi everyone. Today we will be discussing active transport and vesicular transport. We'll start with active transport. Active transport occurs against gradient, which means uphill. So it is an active process. It needs ATP and a carrier protein. For simplicity, imagine someone going up a flight of stairs. He needs energy. Active transport is classified according to which substance utilizes ATP into two types. Primary active transport, which the substance is directly linked to ATP, for example, sodium-potassium pump. Secondary active transport, also known as symport or co-transport. The substance is not linked to ATP but is attached via the protein carrier to the substance linked to ATP, for example, glucose. Notice that sym means connected and co means with. To make it simple, imagine a train pulling a cart uphill. Both need energy to ascend uphill. In this example, the train represents the primary active substance which consumes the energy, and the cart represents the secondary active substance which does not consume energy directly, but it depends on energy consumed by the train. The chain connecting the train to the cart represents the carrier protein. Note, if the train stops ascending, the cart will also stop. So, blocking the primary active transport will in turn block the secondary active transport. Active transport is also classified according to the carrier protein into three types. Uniport, in which the carrier protein carries one substance in one direction, for example, the calcium pump. Symport, in which the carrier protein carries two substances in one direction, for example, the sodium glucose transport. And antiport, in which the carrier protein carries two substances in two opposite directions, for example, the sodium-potassium pump. The sodium-potassium pump. Sodium ions are the main extracellular cations. Potassium ions are the main intracellular cations. This ionic distribution is kept by the sodium-potassium pump. Normally, some sodium ions enter and some potassium ions exit the cells. The sodium-potassium pump functions to bring sodium and potassium to their normal original locations, which means sodium is on the outside and potassium is in the inside of the cells. The sodium-potassium pump is present in most body cells. It is an example of antiport. The carrier protein carries sodium and potassium in two opposite directions. It needs ATP and a carrier protein, which is formed of alpha and beta subunits. The alpha subunit has 3 binding sites for sodium on its inner part, and 2 binding sites for potassium on its outer part. The beta subunit has ATPase activity. When all binding sites are saturated, the ATPase is activated and energy is liberated. This energy causes configuration change in the carrier protein to get the sodium outside and potassium inside the cells. The functions of the sodium-potassium pump. It has 2 functions. Number 1, it keeps the sodium outside and the potassium inside the cells. So, it keeps the normal distribution of ions. And secondly, it is the second cause of resting membrane potential, as it is an electrogenic pump. Because it pumps 3 sodium ions outside and 2 potassium ions inside the cell membrane, it will create a potential difference across the cell membrane, with its outer surface more positively charged. Note, if the sodium-potassium pump is blocked, more sodium accumulates inside the cells and pulls water by osmosis, leading to the rupture of body cells. Vesicular transport. Vesicular transport transports large molecules or particles to the inside or outside of cells. It is an active process as it needs ATP. It is classified into exocytosis, which transports substances outside the cells, and endocytosis, which transports substances inside the cells. Here's the mechanism. Substances get in contact with the coated pit of cell membrane. Then the membrane is invaginated. The membrane then breaks and fuses to form a vesicle surrounding the engulfed substance. The substance is then digested by the hydrolytic enzymes of lysosomes. The good products diffuse into the cytoplasm, and the residue is removed outside the cells by exocytosis. Endocytosis involves two types, phagocytosis and pinocytosis. We will compare and contrast. Phagocytosis deals with large particles, like bacteria and dead tissue, while pinocytosis deals with large molecules, like protein molecules. In phagocytosis, substances are not in solution, which means that they can be seen through a microscope, while in pinocytosis, substances are in solution, which means that they cannot be seen by a microscope. Phagocytosis occurs in phagocytic cells, for example, white blood cells, while pinocytosis occurs in all cells. Phagocytosis means cell eating, while pinocytosis means cell drinking. This is because the engulfed substance is not seen. We only see cells taking water. Vesicular transport is also classified into constitutional, which is non-specific and slower, and receptor-mediated, in which specific substances, for example protein hormones, are attached to specific receptors. It is more rapid. Remember, active transport occurs against gradients. It needs ATP and the carrier protein. In primary active transport, not secondary, the transported substance is linked to the ATP. Carrier protein may be uniport, symport, or antiport. Vesicular transport occurs for large particles, like bacteria, as in phagocytosis, or large molecules, for example protein molecules, as in pinocytosis. Vesicular transport is an active process. It is classified into endocytosis and exocytosis, or constitutional and receptor-mediated. Thank you for watching. Don't forget to like and subscribe, and have a great day.